Hello everyone, and welcome to this audio podcast in this series in line to the LabZtorm effect v.3. This is an audio podcast aligned to storm robotics, a soliloquy, and in and around the role of endogenous opioids in ischaemic preconditioning or IPC in humans. This audio podcast is in around the series. This audio podcast series is aligned to another Storm Robotics production, and this is an audio podcast in and around the mechanisms of ischemic preconditioning or IPC. So, sort of to begin with, in around the mechanisms, if you like, the IPC stimulus has been documented and evidenced to have shown to induce two distinct windows of cardioprotection. The first window occurs immediately after IPC stimulus and lasts up between 2 to 3 hours, which has been referred to as the sort of the classical IPC or an acute IPC, after which effect wanes or sort of disappears or dissipates. The second window follows sort of 12 to 24 hours later and lasts between 48 and 72 hours. And wherein this has been referred to and documented as sort of delayed IPC or second window of protection, also referred to as SWOP, SWOP. And despite intensive investigation, the actual mechanisms that mediate this cardioprotective event remain incompletely appreciated. Although there have been various signaling pathways underlying IPCs that have been documented and evidenced in the literature, only a very simplified sort of very simplified overview has been sort of provided and documented in a line to various research and sort of laboratory sort of investigations. In brief, the IPC stimulus is made up of cycles of brief ischemia and reperfusion and initiates production of a number of autocoids such as and for example acetylcholine, adenosine, bradykinin, endothelin, and opioids by cardiomyocytes. As these autocoids bind respective receptors on the plasma membrane of cardiomyocytes to stimulate several signaling pathways that convey a cardioprotective signal to mitochondria. In mitochondria, signaling reactive oxygen species or ROS are generated and activate protein kinases such as AKT, ERK half or protein kinase C, and tyrosine kinase, which provide sort of the mechanistics around the memory. This process allows the cardioprotective effect to last up to again 2 to 3 hours in what has been documented as classical IPC. At the same time, the mechanisms involved have been documented where for example there is evidence indicating that neuropeptides, for example calcitonin gene related peptide, bradykinin, and neurotransmitters, so for example norepinephrine, precipitate the classical IPC through the participate in the classical IPC, although the precise mechanisms involved in IPC have not been completely sort of distinguished or identified or even established. It promotes IPC in around the mechanisms or mechanistics. IPC promotes the accumulation of cardioprotection ligands or triggers, such as again adenosine, bradykinin, opioid peptides, and prostaglandins. The coupled to protein kinase C or PKC and activate a these coupled to protein kinase C or PKC and activate a number several mediators, such as tyrosine kinase and phosphatidylate in cytol 3 kinase or PI3K, AKT, and P38 mitogen activated protein kinase or MAPK, extracellular receptor kinase or ERK, and JAK, and STAT or STAT. Once KT is activated, it phosphorylates enos or ENOS to produce nitrous oxide. Nitrous oxide then triggers soluble granulate cyclase to generate cyclic guanosine monophosphate or CGMP, which accumulates and activates CGMP dependent protein kinase or PKG. Kinases are then activated during Ischaemic post-conditioning or IPC, and the evidence has informed that they are all phosphorylated, translocated, or show increased activity in the PC myocardium. Furthermore, when activity is pharmacologically withdrawn, the protection or cardioprotection induced by IPC is also abolished. And if it's not abolished, it is blunted. And so, in addition, some downstream apoptic proteins are inactivated, and these include glycogen synthase 3 beta and BCL2 proteins. Kinases then act on the end effectors, possibly the mitochondrial. So, for example, where potassium or KATP channels may be involved or are affected to prevent opening the mitochondrial transition pore. It was works by Liu and team in 1991, who investigated initially the effect of pre-treatment on infarct size with two adenosine receptor antagonists in both control and IPC in situ rabbit hearts. The team portrayed that adenosine released during PC or post-conditioning occlusion stimulates cardiac A1 receptors, which leaves the heart or cardiac muscle protected against infarction even after adenosine was or had been withdrawn. Both adenosine and opioids or GPCR families, where GPCR families are involved, have been demonstrated and evidence to provide significant protection against ischaemic injury in the myocardium, and also the central nervous system. And at the same time, MK sort of particular ions and channels in and around ATP also play a role in the protection or cardioprotection afforded by ischemic post-conditioning. Normally ischemic in and around ischemic in and around ischemic post conditioning. Normally these channels are inhibited by physiological levels of ATP and are therefore minimally active. Opening of these particular ATP channels during stress, for example, such as ischemia, where there's in and around ischemia and hypoxia, allows potassium ions to enter the mitochondria down a favorable electrochemical gradient. This then allows the intramitochondrial potassium ions to be exchanged for extra mitochondrial hydrogen ions. Works by Gross in 2006 have reported some content in around relating on regarding ligand triggers of classical PC and POC, and this team had informed in genetically modified mice or murine laboratory models. Evidence suggests that A1 and A3 receptors may contribute to cardioprotection, since overexpression of either receptors improve functional recovery from ischemia in transgenic murine laboratory models. And at the same time, in sort of mice and murine laboratory models, it appears that even adenosine administration enhances cardioprotection when administered in reperfusion, more so than administration just before ischemia. Adenosine is released during ischemia and reperfusion, with blood and interstitial concentrations elevated after ischemic injury. The mechanisms of ischemic preconditioning the mechanisms in and around ischemic preconditioning are multifactorial, and the exact relationship or association between the various signals is not completely been has not been comprehended. Tuber necrosis factor alpha or TNF alpha and other triggers or mediators which may initiate the cascade of conditioning produce a PC-like effect. And TNF alpha or tuber necrosis factor alpha plays an important role in post-ischemic injury to various organs, and it appears that a non-specific innate immunity may be involved in ischemic preconditioning, and the main elements of the process are, for example, where there's gene expression, leukocytes, and mitochondria. Less has been known in and around the mechanisms or mechanistics underlying remote ischemic preconditioning. So that now draws this audio podcast to a close. The next this has been an audio podcast in and around LabZtorm effect, v.3, and in and around the role of endogenous opioids in ischaemic preconditioning IPC in humans, and an audio podcast series aligned to a storm robotic soliloquy, and where this audio podcast has been in and around the mechanisms of IPC. This has been another Storm Robotics production. In following this particular audio podcast, there will be an audio podcast sort of focus on another aspect of practice, if you like, practice and aspects of research, before sort of a continuation into sort of part two of this series aligned to the role of endogenous opioids in ischaemic preconditioning, or IPC, in humans. The next audio podcast in this series, or where there will be a continuation, will be in line the mechanisms or the mechanistics of remote ischemic preconditioning, or RIPC. So thank you for tuning in on the audio podcast in this series, the LabZtorm Effect v3 thus far, and hopefully listeners can catch up on the audio podcast in around this series sort of subsequently, and wherein there will be a sort of a more sort of in-depth sort of link into aspects of this subject entity. So thank you for tuning in, and thank you for tuning in, and hopefully listeners can check out this audio podcast, the sort of subsequent audio podcast in this series in due time. Thank you.